Right now, inside hospitals across the world, a silent war is being lost. Bacteria that laughed off our strongest antibiotics just killed someone. In fact, by the time I finish this sentence, antibiotic-resistant superbugs will have claimed another life. 1.27 million people every year. More than HIV, more than malaria. But here's where it gets interesting. Scientists have discovered that bacteria themselves invented the ultimate weapon against each other billions of years ago. And we just figured out how to steal it. This is the story of CRISPR, nature's original smart missile system, and how it might be the only thing standing between us and a post-antibiotic apocalypse. But before we get to the solution, you need to understand just how badly we're losing this war. See, when Alexander Fleming discovered penicillin in 1928, we thought we'd won. Bacterial infections that killed millions? Handled. Surgeries that were basically death sentences? Now routine. We got cocky. And bacteria? They got busy. Here's what nobody told you in biology class. Bacteria are the ultimate survivors. They've been around for 3.5 billion years. They've survived asteroid impacts, ice ages, and literally everything Earth has thrown at them. You think our antibiotics scared them? They adapted. Every time we deployed a new antibiotic, bacteria treated it like a training exercise. The survivors passed their resistance tricks to the next generation. But here's the terrifying part. They didn't just pass it to their children. They shared it with strangers. Bacteria have these things called plasmids, tiny circular pieces of DNA that work like USB drives. One bacterium figures out how to beat an antibiotic, and it can literally hand that genetic cheat code to a completely unrelated bacterium sitting next to it. Imagine if you could hand your immunity to the flu to the person sitting across from you on the subway. That's what bacteria do every single day. And now, we're running out of antibiotics that work. The World Health Organization has a list. They call it the priority pathogen list. Basically, it's a hit list of bacteria that we're losing to. Names like MRSA, CRE, drug-resistant tuberculosis. By 2050, that's just 25 years from now, antibiotic resistance could kill 10 million people annually. That's more than cancer, and it could cost the global economy $100 trillion. We need a new weapon, something these bacteria have never seen before. Except, what if the weapon they've never seen is actually the oldest weapon in existence? Now, here's where things get wild. Billions of years before humans existed, bacteria were already fighting their own war against viruses. Specifically, against bacteriophages, viruses that do nothing but hunt and kill bacteria. And in this ancient arms race, bacteria evolved something incredible, a genetic immune system that could remember viral attacks and destroy invaders with surgical precision. They called it CRISPR, clustered regularly interspaced, short palindromic repeats. I know, scientists are terrible at naming things. But what CRISPR does is absolutely brilliant. Picture this. A bacterium gets attacked by a virus but survives. It takes a piece of that virus's genetic code and stores it in its own DNA, like keeping a mugshot in a filing cabinet. The next time that virus shows up, the bacterium recognizes it instantly and deploys a molecular assassin called Cas to cut the viral DNA into pieces. This is not some lab creation. This is evolution's answer to biological warfare, refined over billions of years. And in 2012, two scientists named Jennifer Doudna and Emmanuel Charpentier figured out how to reprogram it. They won the Nobel Prize for this, and they deserve it, because what they discovered was that the mugshot the CRISPR system uses to find its target, it's just 20 letters long, 20 nucleotides, the genetic equivalent of a 20-character address. Change those 20 letters, and you can target anything. Any gene, any organism, any bacteria, including superbugs. Now pause for a second. Let me make sure you understand how precise this is. Traditional antibiotics are like carpet bombing a city to kill one terrorist. You drop the bomb, and sure, maybe you get the bad guy, but you also destroy everything else. Your gut contains 100 trillion bacteria from over 1,000 different species. Most of them are helping you. They digest your food, train your immune system, even affect your mood. When you take a broad-spectrum antibiotic, you kill 99% of them, indiscriminately. And that's why you feel awful. That's why you get secondary infections. That's why antibiotic-associated diarrhea is a thing. CRISPR is different. CRISPR is a sniper. You can program it with that 20-letter address, and it will find only the bacteria carrying that specific genetic code. It can hunt down only the superbugs, only the resistant strains, and leave everything else untouched. But wait, it gets even better. Remember those plasmids I mentioned? Those USB drives that bacteria use to share resistance? CRISPR can target those specifically, Scientists have designed CRISPR systems that don't even kill the bacteria. They just find the plasmid carrying the resistance gene and shred it. The bacterium survives, but it's been disarmed. It's like taking the gun away from a soldier instead of killing them. And because you're not killing the bacterium, there's less evolutionary pressure to develop resistance to the CRISPR system itself. This is surgical genetic warfare at the molecular level. But here's the question everyone asks. How do you actually get CRISPR inside the bacteria you want to target? And this is where the story comes full circle. Because to deliver CRISPR into superbugs, scientists are using the very thing CRISPR was originally designed to fight. Bacteriophages, bacterial viruses, the natural predators that have been hunting bacteria since the beginning of life. Think about this for a second. We're taking bacteria's own immune system, loading it into bacteria's worst nightmare, and using it to hunt specific bacterial targets. We're turning bacteria's own weapons against them, delivered by their oldest enemy. This is some Inception-level biological irony. Here's how it works in practice. Scientists engineer a bacteriophage. These things look like tiny alien spiders, by the way, absolutely nightmare fuel. And they pack it with a CRISPR system programmed to target a specific resistance gene. Let's say NDM1, one of the nastiest resistance genes out there. The phage does what phages do. It hunts down bacteria it can infect. It lands on a bacterium, injects its genetic payload, and now the CRISPR system is inside. If that bacterium is carrying NDM1, the CRISPR system finds it and cuts it. The bacterium either dies from the damage, or it survives but loses its resistance. Either way, we win. And because we're using phages that already know how to target specific bacterial species, we get even more precision. Phages are picky eaters. They usually only infect 1 or 2 bacterial species. Pair that with CRISPR's genetic targeting, and you've got a double filter system that makes friendly fire almost impossible. But wait, this isn't just theoretical anymore. This is happening. In 2023, a company called Locus Biosciences completed phase 1b clinical trials for a CRISPR-enhanced phage therapy targeting recurrent urinary tract infections caused by E. coli. Real patients, real results. Another company, SNP or Biome, raised $60 million to develop what they call precision antimicrobials, CRISPR systems that eliminate specific pathogens while preserving microbiome diversity. This isn't science fiction. This is the future of medicine being built right now. Now, I need to tell you about the different CRISPR systems because not all molecular scissors are created equal. You've probably heard of CRISPR, Cas9. That's the original, the Nobel Prize winner. Cas9 makes clean cuts in DNA. Snip like scissors. It's precise, it's elegant, and it works. But for killing bacteria, scientists have found something even better. CRISPR, Cas3. If Cas9 is scissors, Cas3 is Pac-Man. Cas3 doesn't just cut the DNA, it chews through it. Once it locks onto a target, it starts shredding, destroying thousands of base pairs of genetic code. It's not subtle, it's not elegant, it's brutal. And that brutality is actually useful. Because when Cas9 makes a clean cut, bacteria can sometimes repair the damage. Their DNA repair machinery stitches things back together, and the bacterium survives. But Cas3? There's nothing left to repair. The DNA is confetti. Game over. For targeting superbugs, Cas3 is the weapon of choice. Now, here's where I have to be honest with you, because this video would be incomplete if I didn't talk about the challenges. CRISPR isn't a magic bullet. There are real obstacles that scientists are working through right now. First, delivery. Getting phages to the site of infection isn't always easy. Some infections are deep in tissues or protected by biofilms, these fortress-like structures that bacteria build around themselves. Phages can't always penetrate. Second, phage resistance. Yes, bacteria could develop resistance to phages too. It's an arms race that's been going on for billions of years, and bacteria have tricks. But this is where CRISPR actually helps, because even if a bacterium develops phage resistance, if the CRISPR payload has already been delivered, the damage is done. Third, regulatory hurdles. The FDA and other regulatory bodies are still figuring out how to evaluate these therapies. They're completely new. They don't fit neatly into existing categories. Clinical trials take time. But here's what gives me hope. The urgency is real, and everyone knows it. The alternative, a world where antibiotics don't work, is too terrifying to accept. Imagine a world where a simple surgery becomes life-threatening again, where a scratch from a rose thorn could kill you, where cancer chemotherapy, which wrecks your immune system, becomes impossible because any infection could be fatal. That's the world we're heading toward if we don't solve this. CRISPR offers a path forward. Not the only path. There are other approaches like entirely new classes of antibiotics, antimicrobial peptides, and phage therapy without CRISPR enhancement. But CRISPR's precision makes it special. Let me paint you a picture of what the future could look like. You go to a hospital with a serious infection. The doctor takes a sample and runs it through rapid genetic sequencing. This already takes less than an hour. They identify the exact bacterium causing your infection and the specific resistance genes it carries. Then, instead of giving you a broad-spectrum antibiotic that nukes your entire microbiome, they give you a cocktail of CRISPR-armed phages specifically programmed for your infection. The phages hunt down only the bacteria making you sick. They deliver their CRISPR payloads. The resistance genes are destroyed. The infection clears, and your gut microbiome completely intact. No digestive problems. No secondary infections. No long-term health consequences from having your internal ecosystem wiped out. This is personalized medicine at the molecular level. It's not science fiction. Clinical trials are happening right now. But I want to leave you with something bigger than just the science. What we're witnessing is a fundamental shift in how we think about fighting disease. For a century, our approach was basically find something that kills bacteria, use a lot of it, and hope for the best. It worked for a while, but it was always a blunt instrument, and now that instrument is failing. CRISPR represents a new philosophy. Precision over power. Intelligence over brute force. Working with evolution instead of against it. We're not trying to kill all bacteria anymore. We're learning to edit the bacterial world, to surgically remove the dangerous elements while preserving the beneficial ones. And here's the poetic part. The weapon we're using? Bacteria invented it themselves. We're just borrowing it. Life has been fighting this war for billions of years. Every attack has been met with a defense. Every defense has been met with a new attack. It's an endless evolutionary arms race. By studying that war, we've discovered tools beyond anything we could have invented from scratch. CRISPR is billions of years more sophisticated than anything in our pharmaceutical arsenal. We're not fighting nature anymore. We're learning from it. And that might be the most important shift of all. So where does this leave us? The superbug crisis is real. 1.27 million dead every year, with 10 million projected by 2050 if we don't act. That's the bad news. The good news? We have a weapon. Actually, we have billions of weapons. Bacteriophages armed with CRISPR, programmable smart missiles that can target exactly what we want to destroy and nothing else. Companies are racing to bring these therapies to market. Clinical trials are advancing. The science is sound. We might actually win this war. But here's the thing about wars. They require attention. They require resources. They require society to decide that this is a priority. The silent war in hospitals around the world doesn't have to be lost. The person dying from antibiotic resistance right now, that doesn't have to be normal. CRISPR offers us a chance to rewrite that future. Literally. If you found this video fascinating, hit that subscribe button because I've got an entire series planned on the cutting edge of biotechnology. Next week, we're diving into something even wilder. Scientists using CRISPR to bring back extinct species. And yes, we're talking about the woolly mammoth. Leave a comment telling me, did you know bacteria had their own immune system before this video? I'm genuinely curious. The war isn't over. But for the first time in decades, we might actually be developing weapons the enemy has never seen. And the enemy? The superbugs? They invented these weapons themselves. They just never expected us to use them.